Okay, now let us see the novel approaches for intraocular delivery. So basically, a nanotechnology base ophthalmic formulations are, are one of the approaches which is currently being pursued for both anterior as well as posterior segment drug delivery. So nanotechnology-based system with an appropriate particle size can be designed to ensure low irritation, adequate bioavailability, and ocular tissue compatibility. Several nanocarriers, such as nanosuspensions, nanoparticles, liposomes, and dendrimers have been developed for ocular drug delivery, as you can see in the figure here. So like I mentioned earlier, many studies focus on the delivery to the anterior eye, but with these advancements in nanotechnology, the delivery to the posterior eye is possible, and which is we can use a non-collinear pathway of drug delivery. So one of the example of nanotechnology system is the nanoparticles, which is a colloidal carriers with a size range of 10 to 100 nanometer, and the ultra small size of nanoparticles leading to low irritation and sustained release property, which consequently avoiding frequent administration of the drugs. Avoiding frequent administration means we'll reduce the potential of the adverse effects of the drugs. However, like aqueous solution, nanoparticles may be eliminated from the precorneal area. Thus, one way to improve this is to develop topical administration nanoparticles with mucoadhesive properties, which is to improve the precorneal residence times. And the drug-loaded nanoparticles can be nanocapsules or nanospheres. In nanocapsules, drug is enclosed inside the polymeric shell, while in nanosphere, drug is uniformly distributed throughout the polymeric matrix. And then we have nanosuspensions, which is a colloidal dispersion of submicron drugs particles stabilized by polymers or by adding some surface active surfactants. It is emerged as a promising strategy for delivery of hydrophobic drugs. For ocular delivery, it provides several advantages such as ease of eye drop formulations, less irritation, increased precorneal residence times, improve the bioavailability of drugs, which are mainly insoluble in the tear fluid. So that's why I mentioned earlier that it is a suitable strategy for the delivery of hydrophobic drugs. So basically the efficacy of nanosuspensions in improving the ocular bioavailability of glucocorticoids has been demonstrated in several research studies, because we know that most of the glucocorticoids, they are not really water soluble, and it is really one of the useful drugs for glaucoma treatment. So since it's not water soluble, so one of the options is to deliver the glucocorticoids in nanosuspensions for the ocular based treatment. And these nanosuspensions can also be incorporated into hydrogels or ocular inserts to achieve the sustained drug release. Next is dendrimers, which are characterized as nano-sized, highly branched, star-shaped polymeric systems. And the terminal functional groups may be utilized to conjugate the targeting moieties. So as you can see in the dendrimer, the terminal functional groups is the one that highlighted in red in color. So we can further conjugate it with targeting moieties to improve its permeation or permeability towards engaging with the corneal tissues. Polyamidoamine, or known as PAMAM, dendrimers are widely employed in ocular drug delivery. So dendrimers are basically one of the being utilized as a carrier systems in the drug delivery, and its selection of so the selection of molecular weight, size, surface charge, molecular geometry, functional groups are critical to deliver the drugs. And the highly branched structure of dendrimers allows incorporation of wide range of drugs, which is including the hydrophobic and also hydrophilic based drug. And then we have liposomes, which is a lipid vesicle with one or more phospholipid bilayers enclosing an aqueous core. The size of liposome usually range from 0.08 to 10 micrometer, but for the small unilamellar vesicles, it can be within 10 to 100 nanometer. For ophthalmic application, liposome represent ideal delivery systems due to excellent biocompatibility, cell membrane-like structure, and ability to encapsulate both hydrophobic and hydrophilic drugs. So for drug delivery to anterior segment of the eye, efforts are mainly put toward improving precorneal residence time by incorporating positively charged lipid or mucoadhesive polymers in liposomes. And it has been shown that the positively charged liposome exhibited better efficacy in ocular delivery than negatively charged and neutral liposomes due to binding with negatively charged of corneal surface. And for the posterior segment delivery, liposome development is more focused towards improving the drug half-life, which is by doing so to lessen the clearance from vitreous humor and to protect labile molecule from degradations and also to provide a sustained drug release. And then we have contact lens and implants. So contact lens, as you all know and aware, it is a thin and curve-shaped plastic disc which are designed to cover cornea. Means you can put and it will be inducedly fit to your cornea of the eye. After application, contact lens adhere to the film of tears over the cornea due to the surface tension. So usually, drug is loaded in contact lens by soaking them in drug solutions. Other than that, to improve drug loading inside the contact lens, particle-laden contact lens and molecular imprinted contact lens have also been developed. In particle-laden contact lens, drug is first entrapped in vesicles such as liposomes, nanoparticles, and nanosuspensions, and then these vesicles are dispersed in the contact lens materials. While for the implants, it is available as bio and non-biodegradable forms. It is quite similar to the one that I mentioned earlier on the inserts. And next, we have microneedles. Microneedles-based technique is an emerging and minimally invasive mode of drug delivery to the posterior ocular tissue. So we can use microneedles to deliver drug to the posterior segment of the eye. Microneedles are custom designed to penetrate only hundreds of microns into sclera so that damage to the deeper ocular tissue can be avoided. So this needle help to deposit drugs or carrier systems into sclera or into the narrow space between sclera and chloroid. And puncturing of sclera and depositing drug solution in sclera facilitate diffusion of drugs into a deeper ocular tissue, choroid, and also the neural retina. And the drugs can be delivered in form of solutions, nanoparticles, and microparticles inside the microneedle. Right? So how it release, how it sustain, it release the drugs, it depends on how the drug is encapsulated in a various type of form in nanoparticles or in microparticles. So I think that's all from me, and thank you very much.